Welcome to Ligase Academy. In this video, we are discussing about DNA replication. Before coming to the topic DNA replication, let me tell you about what is a DNA. DNA is the short form for deoxyribonucleic acid, is the molecule that carries genetic information for the development and functioning of an organism. It is made of two linked strands that wind around each other to resemble a twisted ladder. It has a shape known as double helix. Each strand has a backbone made of alternating sugar, that is deoxyribose, and phosphate groups. Attached to each sugar is one of four bases: adenine, cytosine, guanine, or thymine. The two strands are connected by chemical bonds between the bases. Adenine bonds with thymine and cytosine bonds with guanine. That's all about DNA. What is replication? It is a biological process of duplicating or producing an exact copy of DNA. So, it is one of the most vital biological processes in all living things. It is a molecular process taking place in dividing cells by which the DNA creates a copy of itself. As we all know, Watson and Crick proposed double helix structure of DNA. Along with it, they also proposed a scheme for DNA replication. They suggested that the two DNA strands would separate and become a template for synthesis of complementary DNA strands. Therefore, each new DNA molecule would have one parental and one new DNA strand. The scheme was called semi-conservative DNA replication. In 1958, Matthew Meselson and Meselson Stahl performed an experiment to prove this model of replication. In their experiment, there were three models of replication: conservative, semi-conservative, and dispersive model of replication. According to conservative model of replication, during replication, the parental DNA acts as a template for daughter strand synthesis, and when the daughter duplexes are formed, one of them inherits 100% of parental DNA and the other one contains nascent DNA. There is 100% conservation of parental DNA in one of the daughter duplexes. Whereas, in semi-conservative model of replication, parental DNA separates during replication, with each strand acting as a template for nascent DNA formation. When daughter duplexes form, they inherit 50% parental DNA and 50% nascent DNA. In the case of dispersive model of replication, parental DNA separates and are fragmentized during replication. Each fragment acts as a template, and when daughter duplexes forms, there is random inheritance of parental DNA between the daughter duplexes. Significance of Meselson Stahl experiment. Their experiment proved that replication is semi-conservative. This is an isopycnic separation, that is, it is a method of separating molecules based on their equilibrating density with a column. Column is a density gradient, that is, an analyte will show mobility until their density become equal to the density of column. There are two ways of centrifugation: sucrose gradient centrifugation and cesium chloride centrifugation. Meselson and Stahl grew E. coli bacteria in a medium of ammonium chloride with heavier nitrogen isotope N15 for several generations. N15 is the heavy isotope of nitrogen. As a result, all newly synthesized DNA had N15, which can be differentiated from normal DNA by centrifugation in a cesium chloride density gradient. The DNA is heavy DNA called HDNA. After several generations, they transfer the cells to a medium containing N14 as nitrogen source. They are lighter isotopes. Newly synthesized biomolecules now incorporate N14, that is light DNA. Observation of the experiment. As the cells multiplied, they collected samples at regular time interval, that is 20 minutes, 40 minutes, 60 minutes, etc. This is because E. coli cells divide every 20 minutes. So they extracted the DNA, and the DNA extracts are added to a solution containing cesium chloride, and ultracentrifugation is carried out for 24 hours. Cesium, with its higher atomic weight, will form a density gradient in the tube with higher density at the bottom and lower density to the top. The DNA will settle to a layer that corresponds to its density. The tube was shown to UV light at 260 nanometer, and the position of DNA are identified by dark bands. So, these are the predictions of the experiment. If the replication was conservative, after first generation, we'll get two bands, one heavy and one light. After second generation, we'll get two bands with thickness of light band increasing. And all the other subsequent generations also we'll get two bands with the thickness of light band increasing. And if the replication was semi-conservative, then after first generation, we'll get a single hybrid band, and after second generation, we'll get two bands with the same thickness, one light band and one hybrid band. All other subsequent generations will get two bands with increasing thickness of light band. If the replication was dispersive, then after first generation, we'll get single hybrid band, and all other subsequent generations will show a single hybrid band with increasing thickness. As the concentration of N14 incorporated increases one generation to the next, the density of hybrid band constantly decreases, and hybrid band will be seen migrating from the bottom towards the top region. Finally, these are the actual observation of the experiment. After first generation, we'll get single hybrid band, and after second generation, we'll get two bands with the same thickness, that is one light band and one heavy band. And all other subsequent generations will give two bands with increasing thickness of light band. Thus, Meselson and Stahl proved that replication is semi-conservative through their experiment. Hope you all understood about today's session. We will discuss prokaryotic as well as eukaryotic replication in the next video. If you like this video, please like, share and subscribe. Like Academy, your educational catalyst. Thank you.